The broadcast is now starting. All attendees are in listen-only mode.
Well, good afternoon. Thanks for joining us. Welcome to today's webinar, Enhancing Antifungal Therapy, a two-step rapid diagnostic using PCR and NGS, hosted by the DARK Report. The DARK Report is the preeminent source of business intelligence for the anatomic pathology and clinical laboratory industry. Published every 3 weeks, the DARK Report is supplemented by weekly reporting delivered to your inbox from the DARK News Daily service. These go-to publications from the DARK Intelligence Group deliver high-level business management and market trend analysis to pathologists and laboratory executives. To subscribe, please visit darkdaily.com to receive cutting-edge clinical laboratory and pathology news and to gain insights on business-critical issues of our industry. My name is Ann Tate at the DARK Intelligence Group, here with Ashley Harris, who is managing the technical aspects of today's webinar. A reminder that the call is being recorded. Attendees will be muted during the webinar, but you can message us at any time with questions, comments, or any needs. Just use the question box of your GoToWebinar control panel, where I'll be monitoring the chat for today's call. We will also have some dedicated time here for live Q&A at the end of the presentation. All right, so our featured speakers today are Dr. Jose Alexander. He is a medical and public health microbiologist. He's a graduate of the Central University of Venezuela and certified by the American Board of Medical Microbiology. He is a fellow of the Canadian College of Microbiologists with additional certification in microbiology, molecular biology, infection control, and epidemiology. As director of the microbiology laboratories at Advent Health Orlando, he has implemented rapid antimicrobial resistance protocols with whole genome sequencing and laboratory optimization through dashboard technology. His current interests include antimicrobial resistance, next generation sequencing, epidemiology, and data-driven optimization. Our other speaker is Dr. Nicholas Sanford, and he's been working as a clinical laboratorian and assistant laboratory director after graduating from Texas Tech University with a BS in plant biotechnology and a PhD in biological sciences, plant biotechnology in 2015. He was professionally certified by Stanford University in genetics and genomics and is a board certified medical affairs specialist. Dr. Sanford's research interests are in molecular assay design and determining the clinical utility of diagnostic assays. So our learning objectives today are understanding the historical shift in viral diagnostics, identifying challenges in current fungal diagnostic practices, exploring the role of molecular testing for fungal infections, and introducing MicroGenDX rapid fungal reflexive NGS. So I think we're ready to start and ready to begin. So Dr. Sanford, you have the virtual floor.
Thank you, Ann, and thank you everyone for joining us this morning. I'd like to start with a brief overview. We'll give you some historical context discussing the shift from viral cultures to molecular testing in clinical labs. And then we'll follow on with current challenges with fungal cultures and talk about some of the limitations and implications of delayed or missed diagnoses. Then we'll make a case for molecular testing and how it can provide faster and more accurate results before we introduce the MicroGenDX rapid fungal reflexive NGS assay, which is a targeted ITS sequencing test for species level identification of fungi. Then I'll turn it over to Dr. Alexander to talk about the clinical impact of timely fungal identification for patients and some of his experience with the MicroGenDX services. If you could go to the next slide, please. So viral cultures were developed in the 1940s, and they involved isolating and growing live viruses in cell lines or animal models. The method allowed for direct observation of viral effects on cells, but requires extensive time and resources. So serological testing emerged as an alternative, relying on antibody detection rather than culturing the virus itself. While faster, serology could be limited in sensitivity and specificity, particularly in the early stages of infection. Next slide, please. So traditionally, diagnosing infection has relied heavily on culture methods. These methods, however, present significant limitations. First and foremost being prolonged turnaround times. Viral and fungal cultures take days to weeks to yield results, and this delay often impacts the timely diagnosis and treatment, increasing the risk to patients. There are also sensitivity issues wherein cultures may fail to detect low-level pathogens, and there are some clinically relevant viruses and fungi that cannot be readily grown in culture. Next slide, please. There are also biohazard and contamination risks associated with culturing live organisms. Handling these pathogens in culture increases the risk to laboratorians, as well as potential for contamination during routine plate readings. These cultures are also labor-intensive and complex. Culturing fungi and viruses requires specialized equipment, extensive lab space, and highly trained personnel. Variations in specimen quality and handling also impacts the reliability of the results. Next slide, please. So what was the impact of viral cultures on clinical care? The limitations of viral cultures really highlighted the need for faster and more accurate diagnostic methods to improve patient care and reduce the diagnostic lag inherent to traditional culture-based methods. Next slide, please. So the transition from viral cultures to molecular diagnostics was driven largely by advancements that addressed critical limitations in speed, sensitivity, and practicality. First and foremost, molecular methods were able to provide more rapid turnaround times. Methods like PCR reduced turnaround time from weeks to hours. Next slide, please. And okay. The molecular methods also provide improved test accuracy with unprecedented sensitivity, which is essential for identifying infections in culture-negative and immunocompromised patients. Also, molecular methods are capable of detecting non-culturable viruses. Many clinically relevant viruses like hepatitis C or HIV are difficult or impossible to culture reliably in routine culture. There's also, molecular methods also reduce the biohazard risk by eliminating the need to handle live viruses. And this also reduces the risk of contamination and infection of lab personnel. And the last big driver here was scalability and automation. Automation reduces human error and labor costs, and it makes this testing more accessible to the patients that need it. So what was the impact of molecular testing on viral infections? Well, first, we're seeing the faster diagnosis, and this allows for early and precise treatment decisions. It allows for accurate pathogen identification. So this is allowing physicians to avoid unnecessary empiric therapies and their associated risks. We also have enhanced outbreak control, as evidenced during a recent COVID-19 pandemic. And these molecular methods enabled rapid identification of the emerging virus and the variants emanating therefrom, supporting faster public health responses. We also have improved sensitivity in immunocompromised patients where early detection is essential for timely intervention, and these methods significantly streamline lab operations, providing for significant cost savings. Next slide, please. So we can take some of these lessons we learned from the shift in the in the virology section of the lab from culture to molecular testing, and maybe try to apply this to the mycology section. Fungal cultures were developed in the early 1900s and played a pivotal role in detecting a range of dermatophytes. And while it's still useful, it has several limitations that look very familiar to the same limitations of viral culture. First and foremost, they are time-consuming. Fungal cultures take days to weeks to grow, and this delays diagnosis, but it takes nearly a month for a negative result. These cultures are labor-intensive. They require specialized media, multiple incubation temperatures and atmospheric conditions, and highly skilled personnel. They also have limited sensitivity. Some fungi are difficult or impossible to grow in culture conditions. We also have the biohazard risks associated with handling live fungal isolates, and this can pose significant risks for a laboratory-associated infection. So what are the outcomes of fungal cultures? If we look at data from a partner 460-bed hospital, It ran nearly 12,000 cultures in 2023. We found that 86% of those cultures were negative. And here I'll remind you, it takes 28 days to report a negative fungal culture. So that's 28 days where we're reading plates. It's 28 days where patients are potentially on empiric, expensive antifungal medications. And so so we can go on to the next slide here. And what this does is it paints a picture of high stakes in fungal diagnostics. So accurate and timely identification of fungal infections is critical, especially in vulnerable patient populations. And some of the key contributing factors include rising rates of fungal infections. We've been seeing changing geographic distribution of the endemic mycoses, but we're also seeing increasing use of immunosuppressive therapies. So this is making fungal infections become more prevalent. We also see high mortality rates in invasive fungal infections. Infections like aspergillosis and candidemia carry mortality rates exceeding 40% if not promptly treated, and delayed or incorrect identification can worsen patient outcomes, as fungi might be resistant to standard therapies. And then we also have a limited treatment window where early intervention is critical for successful treatment outcomes, because delayed diagnosis can be the difference between a treatable condition and a life-threatening systemic infection. And lastly, there's a significant impact on the healthcare cost and length of stay. Fungal infections contribute to longer hospital stays, intensive care requirements, and increased healthcare costs due to the need for complex treatment regimens. So this is where I'd like to introduce next-gen sequencing, also called NGS. This molecular technique sequences millions of DNA fragments simultaneously, providing a comprehensive analysis of the genetic material in a sample. This technology allows for the identification of pathogens with high sensitivity, even at low abundance, and has applications in identifying bacteria, viruses, fungi, and parasites. One of the major advantages over traditional culture, which can take days to weeks and may not grow all pathogens, is that NGS detects unique DNA or RNA sequences, which makes it suitable for non-culturable and slow-growing organisms. This speeds up the diagnostic processes significantly, especially for fungal infections. So by targeting specific genetic regions like the internal transcribed spacers in the rRNA genes of fungal genomes, NGS can identify a broad range of fungal species. This is particularly useful diagnosing infections in immunocompromised patients or cases where the fungus is resistant to common or empiric antifungals. Capable of handling complex polymicrobial and biofilm-associated infections, NGS is becoming increasingly recognized as the future of infectious disease diagnostics. There's been a lot of research done on the clinical utility of next-gen sequencing, so much so that the Infectious Diseases Society of America is now recommending NGS as a first-line diagnostic for fungal infections. Its utility in routine and complex cases has the potential to redefine the standard of care in clinical microbiology. So now I'd like to introduce the MicroGenDX fungal reflexive NGS. As I mentioned earlier, this is an ITS targeted sequencing assay, and it begins with a PCR step. Now, when that PCR is positive, which is about 13% of cases, we're going to go ahead and send out a preliminary report that says that we've identified some fungal DNA and we're going to reflex this sample to NGS. You'll see the same report if you get an indeterminate sample, which happens about 7% of the time, and that's when we see a weak or a very weak band on the gel that you see at the bottom of the slide there. The other 80% of the time in our laboratory, these samples are going to be negative. So we can issue that final negative report within 24 hours of sample receipt, which significantly cuts down the 28 days required for a negative fungal culture. And this could be, a game changer in terms of de-escalating empiric antifungals. So if we could throw up the first example report. Now, this is an example of a negative sample. It shows you that no fungal DNA was detected and fungal NGS is not indicated. On the next report, we have an example of an indeterminate sample. So this happened about 7% of cases, and they will there will always be reflex to the NGS because about 50% of the time we can identify a fungal species in these indeterminate samples. And in the last scenario, this is the final positive report. You'll see fungal DNA detected, and then you'll see the genus and species of what was found in the sample. Next slide, please. So the MicroGenDX fungal reflexive NGS can help enhance your capacity and promote antifungal stewardship because of its high sensitivity and specificity for fungal detection, which is greater than 90%. It's got a negative predictive value greater than 89%. So it has high specificity for negative fungal results within 24 hours instead of 28 days. So this is very valuable for antifungal therapy de-escalation. And when you do have a positive result, we can give you accurate speciation in 3 to 5 days, which is an improvement on mold identifications, especially, which can take anywhere from 9 to 21 days, depending on whether you're doing it in-house or send out. We can go to the next slide. And here, I'll turn it over to Dr. Alexander to talk about his experience with these tests.
Thank you, Nick. So as a medical microbiologist and technical director for a microbiology laboratory, many of you also have the same experience that I have when the biology department moved from a culture-based method to actual PCR, molecular diagnostics. And that was a significant change. We used to wait days or even weeks for a respiratory sample for specific respiratory virus. So one of the important with that process was that identification was a critical aspect for clinical management. A similar approach can be done for mycology. If we go back to the turnaround time, we know that after 28 days, based on our requirements, this culture needs to be held in a laboratory. We have a positive fungal culture around only 14%. And this is a significant number because we have a great number of cultures that are negative, and that makes the ideal process for integrating a screening protocol. 14 days for a positive fungal culture, including in average with mold, 5 days for yeast, 9 days for mold only when it's done in-house, or when it's sent out to 21 days. Some laboratories have different setups. Some of them don't do any fungal culture at all. They just send it out for a reference laboratory. But it still is a culture that has to be done in 28 days. They have to be done. Other laboratories are able to do the culture in-house, but they need to be screened weekly. And when there is a positive, send out for identification. The identification is challenged, and that's why the diminished expertise that we currently have in the laboratory. Really difficult to find microbiologists that have experience in mycology. Some of them have to be trained, and some of the people that have been working in mycology, they're also retiring. So we have this conundrum in the microbiology laboratory. There is a cost pressure. We know that even when a few plates are set up and the cultures are only read on a weekly basis, there is a cost added. They have a specialized plates. There is a time dedicated to evaluating these plates to be able to look for fungi, or if it's positive for yeast, and most of them are, to be able to isolate. So there is a cost added in labor. The long turnaround time and the complex process are there. So how do we effectively support the antifungal stewardship? Because this is another important factor for the antimicrobial stewardship. How the laboratory can provide rapid diagnostic for antimicrobial stewardship, be able to act in antifungal therapy. The next slide. And how Nick mentioned before, especially with the survey, so we can see that up to 14% of the cultures are positive. 95% of the positives are actually yeast, and we know that that's the most typical fungal that we found. But also they can be found with bacteria, so they can be worked on the bacteriology bench. In the mold situation, we have up to 20% of molds, and I think that that's why normally we consider the fungal culture specifically for mold. And you can use this data and compare against your own experience and your own prevalence. But we also have to see the cost. And negative fungal culture have an average of $50. 53 different yeast, and you use the multi-top, or for example, a platform and other biochemical platform identification can go up to $63. And 72 if it's a mold identification, multiple plates, and a specific process, microscopy evaluation, labor have to be involved. And if it's sent out, can go all the way up to $380. plus the days that the identification normally is. So based on all this approach, we can see how the fungal can be the next department on the microbiology lab to become completely molecular based. So when we talk about molds, most of the molds, 95, 96% of the molds, identification is probably the last process that is done through the steps of fungal culture. Most of them don't require susceptibility, antifungal susceptibility. Some of the antifungal susceptibility are typically required. But the other challenge is that most of the identification are typical into the Aspergillus fumigatus complex. So there is a challenge for identifying species of fungal beyond the genus. And this is another opportunity for molecular identification, especially the NGS, to be able to expand and being able to offer a better identification for antifungal therapy approach. The next slide. So what is this double step involved? Well, the PCR is the first step, and it's a rapid molecular test for identification of fungal. So we'll be able to say in 24 hours there is a fungal DNA or not. If it's not, 86% of the cases. So in this particular case, the escalation of management can be adjusted in this case. And it's positive. 3 days later or 2.5 days later, the identification will be done with NGS that is accurate and is also more reliable, to the point that we're going to be able to get a full genus and species identification. That is critical for antifungal management. This can be done directly from patient sample, and also there is a high negative predictive value for the 24-hour turnaround time of the PCR. So this is an approach that we can actually see the transition from fungal culture to fungal molecular diagnostic, just like the biology department did around 10 years ago. The next one. So when we see this tool, this is an actual antifungal stewardship tool. If there is a suspected of fungal infection, antifungals are initiated, and there is a suspected or not or unknown area of potential infection, this test can be done from multiple sources. And as Nick will describe later, if there is a known site of infection, a sample can be sent for a rapid fungal PCR NGS. If there is no suspected sources, so they can be done directly from blood. So this is an ideal approach because this can be attached to the initiation of empirical antifungal therapies. And this can drive the de-escalation or escalation of the antifungal based on the final result. The escalation can happen in 24 hours, or escalation and optimization of therapy can happen in around 3.5 days when the DGS is done. So this is antifungal stewardship tool. And especially in units as ICU, where agents as microfungi is routinely used. Other tools some other tool, rapid diagnostic tool, can be done in blood. But one of the advantages with molecular diagnostic is that we can get a rapid identification with no limitation in the amount of organism, but also no limitation or interference with antibiotic with other methods that exist in the mouth. The next one. So this is the full fungal culture workflow. And we can see that these resources can be set up in the plates, and then 28 days, the incubation with a weekly read after the growth is there. And now we have up to 2 weeks of identification using microscopy. It's high technical expertise is required, and sometimes it's challenging to find that expertise. So some laboratories cannot identify this in-house, and this has to be sent out to a reference laboratory. The reference laboratory needs to get the sample, need to grow a fresh culture that will take a few more days. So this is one of the approaches that can be changed. The next slide. So you see the process with the PCR NGS. The sample is prepared, the PCR is done, and at the moment that the PCR result is in 24 hours, there is a completely rule out of a DNA, fungal DNA. And this is achieved thanks to the high negative predictive value. The NGS, if it's positive, is performed, and in 3.5 days, a full identification of them. So in less than a week, the patient can be escalated or de-escalated, or the treatment can be fully optimized. The next slide. So This is an example for the two different perspectives. From culture on the right, a 1,680 positive fungal culture, and in the left, 58,000 positive fungal detection by NGS based on the microGenDX. If you see on the right side, you see that most of the organisms identified is used, and they are the most prevalent ones. But in the mold species, you can see that Fumigatus, Penicillium, Niger, and molds, that is typically filamentous mold, as reported by some reference laboratories, they are not clinically significant. And this is a bias based on the expertise of our laboratory. So technically, we are able to identify some of the mold based on the Fumigatus of Penicillium genus. So when we see the perspective from the NGS, the candidate is still prevalent, but we see other molds add to the species identification, Alternaria, Clavifolium, Pulvularia, Rhodotonium. So we can see that the NGS will allow to identify more species that we'll be able to identify by cancer. The next slide. So there is another opportunity for those laboratories that is able to grow the culture, but they are not able to identify the fungal in-house they send out. That is the NGS solution for microbiology laboratories that is intended for identification of fungi directly from the plate. This is a rapid fungal identification, takes 3 days, but it can be also done for a positive blood culture when you have the mold growing in place. So this can be sent to MicroGenDX, and they can be identified directly from the plate if you don't have the technical expertise to identify these organisms. So this still can do the culture, but it can be found for identification. The next one. So this is an example, and this is another opportunity for NGS. So this is a plate that we have multiple organisms, and we have a Aspergillus, but we also have a white mold. But we're not able to separate that. After multiple tries to separate these two molds, we sent to a reference lab, but the requirement is to have a pure culture was challenged. So the whole plate was sent to MicroGenDX to identify anything that is precious on the shelf. So the Aspergillus fumigatus was identified, and we saw, but then we see the Lactobacillus that was identified, that is the white mold that was present. So we were able to separate both of them using NGS. Besides that, we also find out it was an Nocardia brasiliensis below the contamination with some of the bacteria. And that Nocardia was able to grow later on culture and be confirmed. So these plates will be impossible for us to send to our reference laboratory because the requirement is to have a few samples. But the challenge to sometimes be able to separate fungi from bacteria or the mold require that the plate can be sent to MicroGenDX for identification. Another important example is some of the Aspergillus can be identified by microscopy to Fumigatus complex. But by NGS, this was a Lentulus. So Aspergillus lentulus, we know that it has a high MIC, especially against azote. So identification of the appropriate species or the species into a complex can provide a lot of antibacterial antifungal insight to be able to adjust, optimize them. In this particular case, azote, that is a typical treatment for Fumigatus, with Lentulus, that's something that has to be reevaluated because these species can be resistant to azote. The next one. They can be done also from paraffin. As a microbiologist, it's typical to get some paraffin blocks, some slides to evaluate and being able to report the presence of fungi, or sometimes even to see if we can identify this. If this is a fungal culture going in the microlab, it still will take some time, but this can be done directly. One of the advantages, you have a positive pretest. You know that it's a fungal that was seen on the paraffin block on the tissue, and that paraffin block can be sent to MicroGenDX for speciation. The next slide. And in this particular scenario that was shown on the slide, you can see the hyphae and the mold is obvious that it's present. But 3 days later, based on NGS, we were able to identify Aspergillus variants. So some pathology service, they can be able to correlate better the information from a direct visualization of the mold in the tissue with the result of the NGS that can be highly specific. The next one.
So I'll take back over.
So now I'm going to pass back to Nick. Thank you.
Thank you. So we've done a lot of work to validate our tests for a wide variety of sample types. We wanted to make it as widely available as possible, so just about any sample you can collect off of a patient, we can test it. For the direct colony testing that Dr. Alexander was mentioning, we've also validated a variety of different media that can be sent in. Those are listed at the bottom of the slide there, so we can take those on plates, slants, or bottles. We do ask that, if you're going to be sending whole blood or CSF, to ship those immediately with molecular testing. Sample quality is really critical for getting a good solid result out. All other sample types are stable at room temperature for 21 days post-collection. But when batching prior to shipping, we do ask that you refrigerate or freeze within 24 hours, except for blood and CSF, which should be shipped immediately. Specimen transport media are only required when submitting stool or respiratory viral specimens. And again, there is a list of all the different media that are validated for the direct colony testing. We can go to the next slide. So I'd like to thank everybody for joining us today and listening to our presentation about the MicroGenDX reflexive fungal NGS. If you'd like more information about these products or other tests available, you can visit microgendx.com or contact our customer service center. My name is Nicholas Sanford. I'm the VP of Medical Affairs at MicroGen. And if you have questions about the technology, I'd be happy to answer those. And if you'd like to be contacted with more information, you can scan the QR code that's up on the screen now.
Thank you, Dr. Alexander and Dr. Sanford, for a thorough overview of this fungal diagnostics and the exciting innovations from MicroGenDX. I think it'll be great for patient diagnosis and treatment, so we really appreciate it. So we are ready to begin a Q&A, and we do have a multi-part questions here. So I'm going to start with the first one. In terms of platforms, is one instrument with X number of samples per day, or do you have your own instrument platform? We'll start with that one first.
So we're a reference laboratory. You'll be sending the samples to us, and we're going to run those on an aluminum IC. Was there? I think that was all for the first part of the question.
Correct, in the first part of it. So then the second part of it is if you have more than one instrument, what is your throughput? What is your highest throughput, I guess?
Well, I don't know that we've maxed out yet. I know we're currently doing around 300 to 400 samples a day, but we can definitely take more.
All right. And just for the audience, how is it priced? And I think you can probably get back to the questioner of this question about pricing, unless you want to share pricing.
Yeah, so this test has been priced as it is kind of a two-step. So if you have a negative final result, the cost for that is going to be $80. And if it's indeterminate or positive and it's going to be reflexed to NGS, it's an additional $150 for a final cost of $230.
All right, great. So then our next question is, I think this has kind of been answered, but we'll say it again. This is a test that can be ordered by physicians as well as labs.
We do have. You want to take it, Dr. Alexander?
No, take it. Sorry.
Okay, so we do have testing options available for physicians to order at the bedside, but the rapid fungal reflexive NGS and the direct colony services that we discussed today are more designed for the microbiology laboratory.
Physicians wanted to order it, they could as well?
They could. These tests can be ordered from our website.
All right. So is this something that should be ordered in place of fungal cultures, or should they start with fungal cultures or at the same time? What's your recommendation?
Dr. Alexander, would you like to tackle that one?
Sure. I think it depends on the level of expertise that you have in your facility. If you don't have a fungal culture, you don't do fungal culture at all. So this test can be replacing the culture, sending the sample for identification. You can aliquot the specimen and hold specimen in case that is positive and additional testing is required. Let's say antifungal is needed for that particular mold. There is probably a small percentage of cases. If you already do the fungal culture in-house, you can continue doing the fungal culture. But as soon as you have a PCR negative in 24 hours, you can decide if you want to discard the plates or still keep the plates. If you keep the plates, you can keep them correlated. There is nothing indicating that the fungal culture and the PCR have to be done at the same time. The other way to do it is if it's positive, you want to keep the plates to be able to grow the organism and save the isolates or for any future test. So I think it can be any approach depending on how your laboratory is set up. And the test can be order directed for clinician, especially in units as ICU, or when there is a high suspicion of fungal infection, because they can allow a rapid 24-hour rule out of the fungal infection at the same time that your micro lab is running the code.
Thank you. Going on to another topic is how often are the results inconclusive?
So in our experience, we see inconclusive results in about 7% of samples. When those are inconclusive, we do reflex all of them to NGS because we found about 50% of the time there is a reportable fungal signal in the sample.
Thank you. And a final question is, are you licensed in California?
We are licensed in all 50 states. So we're even licensed in New York State.
All right, great. That's great. Any more questions from the audience? All right, if not, you'll have a chance to pose them to MicroGenDX at a later time. So we've come to the end of our webinar today, and we wanted to thank MicroGenDX for making this possible. And we want to thank our speakers, Dr. Alexander and Dr. Sanford. And we hope that you found this program and this webinar beneficial to understanding NGS and PCR testing, and maybe the gold standard for the future in infectious disease diagnostics. Please be on the lookout for an email with your on-demand link for this program. This will be your chance to ask any additional questions. You still have another chance, and ask for copies of the slide deck. So also remember to check out darkdaily.com, where you can opt in to receive the latest news and program updates. So we've come to the end of our program today, and we thank you for all attending, and I hope you guys all have a great day. Thank you again.
Thank you all.
Thank you, everyone.
Thanks.